Hello. Live? Ravi, live? Yes. Good morning, all of you. How was your first exam? Did you write the exam? Okay, done. Good morning. How was your exam? Okay, done. Practice well. Grow day by day, day by day. After the exam, take your question paper. Keep with that question paper your time for a while. Whatever the questions that you have done wrong, collect those questions, analyze those questions, why this question has got wrong. That's how to grow. Be careful. There is a pattern that will make you successful. There are three parts are there in the practice. One is studying, one criteria. Writing exam, another criteria. After writing exam, correction of the paper. See, one question is there that you have committed wrong. First, you have to understand what is the reason behind the mistake. Reason means, now first thing, did I study this concept? Yes, I studied. Why it got wrong? Because when I was studying this concept, I did not pay attention. Okay, done. If my mind is not paying attention on this area, it might be paying attention somewhere else, right? Where it was at the time of reading this concept. One or two times check, then you will get the clarity. Same mistake you should not do twice. That is the way to grow. Do a mistake. You are eligible to do the mistake. You have every right to do the mistake. But you don't have any rights to do same mistake again and again. Do one thing. If anything that I didn't teach and you have got in the exam, there blame me. If I have taught the thing and you did not answer it, blame will go on you. This mutual understanding we should be having. I will be make sure that there will be no word from the NCERT book you will miss out. You can understand when we going these 3 to 4 classes, you can catch very clearly. I maximally concentrate on NCERT content. And also, if I feel there is a word in NCERT book, that word you may not understand meaningfully. I am taking an example and I am exemplifying the meaning of the word also. So, follow the thing perfectly. One thing, you don't get marks, you have to gain them. Your practice regularly will make you to get the marks. They will not come to you. Okay? Learn the subject, correct the subject, make the mistakes, don't repeat the mistakes, and this is a progressive way. Never think you never do mistake. You will do mistakes, but stay with your mistakes. Don't blame anyone. Stay with your mistakes. Collect the mistakes. Why did I do this mistake? What could be the reason behind it? Okay? If I studied and I am doing wrong, where the things are going wrong, analyze yourself. Improve your practice. regularly make the things rectified, not one day, regularly. Okay, sudden changes never ever going to occur. Sudden changes will never stay longer. One day, if you study 8 hours, you may be sitting due to some sort of, sudden motivation, but how long it will stay? That is a difficult thing. If you have a habit of sitting and studying regularly for 5 hours, regularly for per day 5 hours you are sitting. One day if you sit 8 or 9, doesn't matter, you can cope up, you can able to develop. But suddenly if you want to sit and study, it is not at all possible. Everything is a practice. One thing, while you are solving one problem, I will give you one hint. You did not get ninth table remembering. They have given one statement in ninth table, you did not remember it. Ninth table you have forgotten, that's okay, agree you have forgotten ninth table. Don't say to anyone, realize you have forgotten the ninth table or you did not learn ninth table. Silently go, sit in a corner and learn the ninth table. Don't say to anyone, develop. That's called development. Suddenly, a god or goddesses will not give marks to you. Marks are the result of your work. Marks are not bestowed or given by somebody. Marks are gain of your work. So believe that. Understand? Somebody saying here some kind of things. How can we get question paper again, because after exam the paper unavailable? That would be if you ask the concerned people who are, they will give it the paper again to you. It's not a big issue. Lateral meristems are cylindrical meristem, is it true or false? Lateral meristems, I think when the class was going on, did I give about this? lateral meristems are Lateral meristems are cylindrical meristems. Yes, I have given. If you check in there, lateral meristems are cylindrical meristem. Okay, then come back. Let me talk about that. Next one, dicot stem. Up to monocot root we have completed. Now I am beginning dicot stem. Dicot stem. Dicot stem. When you talk about dicot stem, first thing, we will take a young dicot stem. Always. An old dicot stem will not have the following features we will talk about. Why? A dicot stem, if it become old, it performs secondary growth. The secondary growth will make most of the primary tissues will be disoriented, gradually disappear. That's why whenever you take anatomy of a plant, especially dicotyledonous plant or monocot, we will use their primary body only. Okay, primary body. So primary dicotyledonous stem choose, and then go for cross cutting, I mean transverse section, and observe from the top. In this area, observe, I will choose here sunflower stem. Sunflower stem, most commonly used one. Let us see one by one. First one, I am taking a patch of dicotyledonous stem and I am analyzing the tissues. A small patch of the dicotyledonous stem. First one, uppermost part is epidermis. Everyone knows epidermis. Epidermis is unilayer or multilayer? Epidermis, unilayer of cells. Okay, they are parenchymatous or collenchymatous. Every time epidermis is made up of parenchymatous cell. So far there is no epidermis other than parenchymatous cell, but typically it is made up of unilayer of parenchymatous cell. Then come back. On the top of the parenchymatous cell, so called epidermis, in the young stems will have a water impervious layer. That impervious layer is known as cuticle. Cuticle could be seen only in case of young stem. If a stem that it has been old, or the stem had performed some kind of secondary growth, cuticle gradually disappear. That's why young stems only will have cuticle. Then these stems, they possess, do you remember what the structures I'm writing? They are called trichomes. One thing, trichomes would be found only in case of dicotyledonous stem. You don't see trichomes in monocotyledonous stem. That's why monocotyledonous stem typically monocotyledonous stem typically are glabrous, which means they have smooth appearance. Dicotyledonous stems are hairy, which means on the top of the stem you can see a lot of hair, but in monocots this hair is absent. But that hair is on the stem technically known as trichome, so that the trichomes may be branched, unbranched. First of all, they are multicellular, and then sometimes soft, stiff, even sometimes secretory in nature. So main work of the trichome is they are reducing transpiration rate by closing some sort of stomata present on the epidermis. That's about trichomes. Then go below epidermis. Below the epidermis, in case of dicotyledonous stem, there is a presence of special type of cells. They are having, okay, corner wise more thickness, and then these cells are living, they do have some sort of mechanical stability. Do you remember what type of cells which are responsible for providing mechanical stability by having chloroplast in them? There is only one layer in the plant body which can provide mechanical stability even they are living. The living mechanical tissue is nothing but hypodermal area made up of cholenchyma. Cholenchyma. Now, when it comes to cholenchyma, the always in an young stem, cholenchymatous tissue is present in the form of a hypodermis. This is not compulsory to have hypodermis cholenchymatous cell. There are here and there exceptions are there, but if it is present, it is present in the form of hypodermis. But one thing, in case of sunflower, this hypodermis is continuous layer, continuous. Whereas in case of cucurbitaceae members, this hypodermis is discontinuous. First, in some cases it is continuous, in some cases it is discontinuous. But wherever you see, the cholenchyma would be present in the form of hypodermis layer in a dicotyledonous stem. Then go to below the hypodermis. There are large number of cells with conspicuous intercellular spaces arranged. These cells are known as parenchymatous ground tissue. I will call it as cortex. Sometimes it is called general cortex, sometimes it is called as cortex, but anyhow they are made up of parenchymatous cell. The major recognition of them is between these cells there is a presence of conspicuous intercellular spaces. Intercellular spaces, they are conspicuous. Large intercellular spaces are present. These large intercellular spaces technically absent in case of collenchyma. They will be present only in case of parenchyma. Got it? Next one. Below the cortex, there is a presence of endodermis. This endodermal layer, it is also made up of barrel-shaped cells. What shape? Barrel-shaped. Barrel-shape means these cells are not having any sort of gap. These barrel-shaped cells will be arranged without any intercellular spaces, but the interesting one, in them you will identify there is a presence of starch grains. What they are? Starch grains. Now, the starch grains containing endodermis is a characteristic feature of dicot stem. That's why this dicot stem endodermis is also known as starch sheath. Starch sheath. Okay, one more thing, you will see in different places there is a presence of endodermis, but that endodermis could be having starch grains. That is a characteristic feature of only dicotyledonous stem. Nowhere else you will see a layer with starch grain which is made up of parenchyma apart from dicotyledonous stem. That's why starch sheath endodermis is a characteristic feature of dicotyledonous stem. Next one, see below that, below the endodermis, whatever the layers of cells are present, they are called stele. This word we have learned. Stele means core, inner side. Whatever the layers that are present below the endodermis, all those layers combinedly are known as stele. Okay, let us see in the first layer of the stele. The first layer of the stele is nothing but semilunar patches of sclerenchymatous cell. The sclerenchymatous cells are arranged like a semilunar patches. These are semilunar patches of sclerenchymatous cell. Semi-lunar patch like sclerenchymatous cell. Sometimes these sclerenchymatous cells which are existing in the form of semilunar patches, they are also known as bundle caps. Why? Below each pericycle layer, one vascular bundle is present. That's why they are called bundle caps. What do you call them? Bundle caps. Bundle cap. Next, this semilunar patch always made up of sclerenchyma. Very, very important. Sclerenchyma. You don't see the sclerenchymatous layer in the dicotyledonous stem elsewhere, or in the entire dicotyledonous stem. You will see only in one place the sclerenchymatous patches are present. The sclerenchymatous patches are nothing but pericycle. Okay, next. Below the pericycle, which is semilunar patch, you will find vascular bundle. Here I have taken two vascular bundles. Now, in this, observe, there is a presence of xylem. Xylem present towards the center. Inside the xylem, protoxylem present towards the center. Metaxylem present towards the periphery. Don't mistake. When you compare between xylem and phloem, phloem could be existed towards the periphery in a vascular bundle. Okay, xylem present towards the center. Now, inside the xylem, if you check vascular bundle arrangement, this is metaxylem, which is present towards the periphery. The small cavity contain protoxylem present towards center. If protoxylem present towards the center, metaxylem present towards the periphery, such kind of arrangement is called, do you remember? Protoxylem center, metaxylem periphery. That arrangement is known as endark protoxylem. Endark protoxylem. Sometimes I have seen just leaving the word called endark xylem, endark xylem, endark. No, no. You have to significantly mention that is endark protoxylem. Sometimes they will also mention it is exarch metaxylem, but it is endark protoxylem. Clarity should be there. Next, go to vascular bundles. This is phloem, primary phloem, and this is primary xylem. Between the primary phloem and primary xylem, we will see there is a presence of a kind of cambium. This cambial tissue is known as intrafascicular cambium. Intrafascicular cambium, or sometimes it is also called vasicular cambium. Intrafascicular cambium or vascular cambium, like this. Xylem, phloem separated by intrafascicular cambium. This kind of vascular bundle is known as, I think you know it already, that kind of vascular bundle is known as conjoint collateral open type. Why did you call this vascular bundle as open? It is open because between them there is an existence of intrafascicular cambium. Okay, like this. You see these two vascular bundle, both are almost similar in size. They have been capped by sclerenchymatous bundle sheath, sclerenchymatous pericycle. That's the reason why they are having, that's the reason why the pericycle is known as bundle cap. So between the two vascular bundle, whatever existing gap is present, that gap is called medullary rays. They have another name called pith rays. Pith is the central portion of stem or any area, pith which is filled with parenchymatous cell, and in case of dicotyledonous stem, you will identify a large pith which is covered by vascular bundles. Now, this is overall structure. Let me take an image that's given in our NCERT book, because in NCERT book, due to high level of printing quality, that image is very clear. Unless you have cleanly understand what are the parts mentioned in what are the area, you cannot able to understand what exactly the diagram is. So I will take the diagram and exemplify what are the parts. Take this NCERT book. We have anatomy of flowering plants, right ma? Anatomy. There this is dicotyledonous stem. Anatomy of dicotyledonous stem. The patch that I have written earlier is nothing but this patch. Let me take the detailed versions of what are the parts they're trying to mention. Let me confirm. First one, this is epidermis. These are trichomes, easy to be understand. Next one, below that there is a presence of a layer of collenchyma, few layers of collenchyma. In fact, this you will call it as hypodermis. Next one, below that there is a presence of parenchyma. This is called general cortex. Okay, endodermis. Observe from endodermis. Endodermis is unilayer, but here there is no unilayer of endodermis clearly mentioned there. Overall, it appear like the endodermis and the pericycle has been mixed up and creating a kind of idli-like structure. That's not actually over there. There should be a clear-cut distinction between the pericycle and the endodermis. That now I am talking about pericycle first. If you see, First endodermis, if you count over there, this is the endodermis. Endodermis has another name. Do you remember what is the another name? Starch sheath. The endodermis is having a special name called starch sheath. That is this starch sheath. Next one. Below the endodermis, there should be a presence of a sclerenchymatous patch. Yes or no? That sclerenchymatous patch usually should be in the form of semilunar patches, half moon shaped patch. That half moon shaped patch is called pericycle. One thing, come to the conclusion. When we were talking about pericycle of dicotyledonous root or monocotyledonous root, do you remember root pericycle made up of thick walled parenchymatous cell? And we have also mentioned this thick walled parenchymatous cell later responsible for the formation of lateral roots. During growth of the root, lateral roots will be formed. These lateral roots are developed from pericycle. That's why I said lateral roots of any plant endogenous, root hairs are exogenous outgrowth, isn't it? Now in that case, here you are talking pericycle. In case of the stem pericycle is dead, root pericycle is living. Stem pericycle cannot grow at all. Root pericycle can grow and form lateral roots. That's a distinction between root pericycle and stem pericycle. Keep it in mind. Then go below that, you will find vascular bundle. This is actually one vascular bundle. One vascular bundle, one bundle. Okay, in one vascular bundle, you know, xylem, phloem, intravascular cambium should be present. Let us see from peripheral to inner what are those parts are present. Next, first phloem. This is primary phloem, you can call. Why I am writing phloem, primary phloem particularly, later you will understand. When we begin the process of secondary growth, this primary phloem, primary xylem, gradually these tissues are started to deteriorate and their importance will be decreased. That's why they could be easily noticeable only in a dicotyledonous stem before the onset of secondary growth. Next one, primary phloem, and then there is a presence of xylem. Xylem towards inner side, phloem present towards the peripheral. Between the xylem and phloem, there should be a presence of cambium. What cambium they are talking about? Intravascular cambium. And then tell me, when I was talking about meristems, I was mentioned this intravascular cambium is primary meristem. Am I right? And also the intravascular cambium, we can also call fascicular vesicular cambium. Fascicle, English term, indicate bundle. The cambium that is present inside the bundles. Who are the bundles here? Obviously our vascular bundle. That's why the vesicular bundle is also known as fascicular bundle. The bundle that is present, I mean the bundles contain some amount of cambium which is primary cambium. That cambium is known as bundle cambium, fascicular cambium, or intravascular cambium because it is present inside the vascular cambium. Later you will see there is an again development of some intervascular cambium for the sake of secondary growth. So since we are talking primary, this is intravascular cambium. Then go to xylem. Xylem, metaxylem, large cavities, protoxylem, small cavities. So protoxylem towards the center, metaxylem present towards periphery. This arrangement is known as endark protoxylem. Endark protoxylem. So below that area is known as pith. Pith is made up of parenchymatous cell. Next, go overall structure. If you notice, this is the body. This is a complete picture of a dicotyledonous young stem. There you see I'm going from peripheral to inner. What are the parts present there? I wanted to notice. I'm going from peripheral to inner. Outermost epidermis. Epidermis will have trichomes. What are these decorations? These are all are trichomes. Okay, that much, you know, elaborately trichomes are not seen, but this is a diagram. They have written trichomes. Remember, that much thickness of trichomes will not be there. Okay. Next one. Below that, epidermis. Below that, hypodermis made up of collenchyma. Below that, general cortex. Below that, endodermis. Endodermis other name is starch sheath. Below that, you will have pericycle. Let us see there. This portion is known as pericycle. Keep it in mind, boys. This is called pericycle. Okay, pericyclar area. Next, below that you will have primary phloem. Below that you will have intravascular cambium. Below that you will have metaxylem, and then finally you will get protoxylem. Why I am sequencing them? Mostly they are framing the questions like this. If we are moving from peripheral to center, what kind of arrangement it is? Centripetal arrangement. When it comes to centripetal arrangement, okay, that is peripheral to center, what is the first kind of xylem will come? What is the first kind of phloem will come? Altogether, you can able to arrange them like this. Next, this is a vascular bundle, another vascular bundle. Between the two vascular bundle, there is an existence of gap. This gap is called medullary race. Medullary race. Gap is known as medullary race. Important. The medullary race sometimes is also called pith race. Then all vascular bundles in the dicot stem almost similar in size. You can see that all of them are similar in size. That similar size vascular bundles all are arranged like a ring. Ring like arrangement of vascular bundle. Ring like arrangement of vascular bundles. Vascular bundles. All vascular bundles I am talking about. Shape of the vascular bundle is not ring. Shape of the vascular bundle is triangular or sometimes wedge shape. Wedge shape. Shape of one vascular bundle. But all those wedges arranged like a ring in the center of the stem. This arrangement is known as eustele. Eustele. Eustele is a characteristic feature to dicotyledonous stem. Very important. In the entire plant anatomy, only two places where we will find this eustele. One is dicotyledonous stem. Another one is called gymnosperm stem. Apart from that, the eustele, which is nothing but all vascular bundles similar in size without, with arrangement of ring. Okay, that arrangement exclusive for two places, dicot stem and also gymnosperm stem. This is eustele. The concept of the eustele again we will take. Okay, when it comes to secondary growth, I will take this idea of eustele one more time. There we will elaborately discuss. But now we will move on to monocotyledonous stem. One more thing, sometimes they will ask centrifugal order from center to periphery. Sometimes they will ask centripetal order from peripheral to center. At the time, which is present where, you should have a clear idea. Epidermis to pith, that is centripetal order. Pith to epidermis. Just one or two times write them in the form of a sequence, you will easily get an idea. It's not a big deal. Got it? Next one, monocot stem. Observe, when we talk about monocotyledonous stem, the story goes like this. Same arrangement with slight variations. Same layers, slight little bit variations are there. First one, they have epidermis. Usually the epidermis is unilayer, but closely observe, when you take dicot stem and monocot stem, something is missing in this ring and that something is present in that ring. Could you say that? What was that? Outer side I am talking about. That should be trichomes. Do you notice that? Trichomes are not found in the monocotyledonous epidermis, but trichomes are seen in dicotyledonous epidermis. Very important. Dicots generally possess trichomes. I am not saying Every dicot definitely should be having trichome. That's another wrong statement. But trichomes are generally could be seen in case of dicotyledonous stem. Monocot stems, no trichomes would be seen. Then come to hypodermis. Hypodermis is very interesting layer in case of monocot stem. It is made up of sclerenchyma. Do you remember that? Where did you see the sclerenchyma in case of our dicot stem? In the entire dicot stem, we have seen sclerenchyma only in one place. What was that? Pericycle. But in case of monocot, they need a little bit more strength because they do not perform secondary growth and they do need some sort of flexibility. Even for the flexibility, there is no existence of collenchyma. That's why in case of monocots, hypodermis made up of sclerenchymatous cell, and the sclerenchymatous hypodermis is dead, but the hypodermis of dicot is living. That's a living mechanical tissue. This is a dead mechanical tissue. Both dicot hypodermis, monocot hypodermis mechanically strong, but the nature of mechanical tissue present over there is different in dicot and monocot stems. Next one, you take after the hypodermis. One thing, some words you need to remember: endodermis, endodermis, pericycle, pericycle, and also medullary rays, medullary rays, etc. are absent in case of, very important, absent in case of monocot stem. You don't see the distinction of endodermis, pericycle, medullary rays, pith. These distinctions could not be clearly seen in case of monocotyledonous stem. The entire area inner side would be filled with special type of large sized parenchymatous cell. That's why we will call them as, okay, there is ground tissue existing, not distinct to ground tissue. One thing, hypodermis is a ground tissue part, general cortex is a ground tissue part, endodermis is a ground tissue part, pericycle is a ground tissue part, and also medullary rays, pith is also ground tissue part. Why I am saying this? When we were talking about different varieties of tissue systems, you said this word, except epidermis and the vascular bundle, remaining entire body of the plant is made up of ground tissue. Am I correct? Now, you are talking about ground tissue word here. The ground tissue, it's not just, it's made up of one layer of cell. Variety of layers will be there. But all these layers are not that much distinct in case of monocot stem. You cannot have distinction of endodermis, general cortex, pericycle, medullary rays, pith. All these distinctions useless in case of monocot stem. Why? The entire inner space of the stem filled with parenchymatous cell. So one word is enough here. That's called ground tissue. Inside the ground tissue, if you go here and there, inside the ground tissue, here and there, you will identify this. Inside the ground tissue, here and there, you will identify some vascular bundles which are scattered. Vascular bundles are scattered. Presence of scattered vascular bundles, technically known as atactostele. Atactostele. Presence of ring-like vascular bundle is eustele. If the vascular bundles are scattered, that's called atactostele. Okay, they are scattered, they are not arranged in the form of a ring. But one thing you observe closely over here, vascular bundles uneven in size. Peripheral vascular bundles are smaller and the central vascular bundles are larger. Very important. So, size of the vascular bundle gradually increases from peripheral to center. Why peripheral vascular bundles are small and central vascular bundles are large? The small peripheral vascular bundles, actually they are immature. They are what? Immature. Immature peripheral vascular bundle. Next one, the central vascular bundles which are large. Large vascular bundles are matured ones. What does it mean by matured? Matured. For what purpose? You are talking them as matured and immature. Remember, matured vascular bundles will have water transporting capacity, water and food as well. But immature vascular bundles will have nothing to be transported. Just now they have formed, they have to attain the ability to transportation in future. Such kind of immature or non-functional vascular bundles could stay periphery, and the remaining vascular bundles which are working actually will be present in the center. That's the reason why central vascular bundles are larger, peripheral vascular bundles are smaller. Peripheral vascular bundles non-functional because of they have immature vascular tissues. Next, central vascular bundles are mature and they have large cavities. They can attain the ability of transportation. This is called peripheral and central vascular bundles, which are dissimilar in size. Then go to one vascular bundle. There are many. I am talking about one vascular bundle. Next, if you take vascular bundle, they are saying this is one vascular bundle, this is another vascular bundle. Like that, many are there. They are scattered, peripheral smaller, central larger. Okay, but observe one vascular bundle, its unique features. If you take a vascular bundle, always vascular bundle covered by a special type of covering known as bundle sheath cells. Bundle sheath. Now, wherever you come across a word called bundle hereafter, bundle means they are talking about vascular bundle. No other bundle exists inside the plant body. That is default consider. Bundle means a bunch of cells. They will be present here and there. But if they are talking about bundle sheath, bundles, first priority of the term bundle is vascular bundle. So there is a vascular bundle. Around the vascular bundle, there is a presence of a sheath of cells. The sheath of cells or covering of the cells is known as bundle sheath. Interestingly, this bundle sheath is made up of sclerenchyma. They are very fond of sclerenchyma. The monocot stems are very fond of sclerenchyma. Why? They want strength. Whatever the strength they can obtain in the primary stage, the same strength will be continued. They can't develop further attainment of mechanical tissues in future. That's the reason in maximum places they try to keep those cells which are providing mechanical strength. Why you are saying that maximum places? Simple. They have already kept one layer of sclerenchymatous cell in the hypodermis. The another layer of sclerenchymatous cells they are keeping around the vascular bundle, that we are calling it as bundle sheath. Very important. Sclerenchymatous bundle sheath is a characteristic feature of monocot stem. Again, there are so many places you will come across existence of bundle sheath. You will see them in leaf, you will see them in different other kind of areas also. But wherever you see the bundle sheath typically parenchymatous. But the only plant which is having the bundle sheath which is sclerenchymatous is monocot. Okay, around the bundle sheath cell there should be around the bundle there should be a presence of sclerenchymatous layer, that we call it as bundle sheath. Next one, go to each vascular bundle. In each vascular bundle they have large metaxylem towards one area, protoxylem towards another area. Same rule, metaxylem towards periphery and the protoxylem towards center. Such arrangement you can call it as endark protoxylem. In case of monocot stem also, protoxylem is endark. Do they have phloem? Of course, if you are saying vascular bundle, xylem and phloem together is known as phloem. But the phloem present towards the peripheral portion. One thing you have to talk about phloem. Come back. When we were talking about angiospermic phloem, what are the tissues that you have denoted present in the phloem? sieve tubes, companion cells, phloem parenchyma, phloem fibers. Am I correct? These are the four types of tissues present in phloem. But on the day, we mention one among the four missing in monocots. Could you tell me which of the four phloem tissues missing in monocots? Which one that? That should be phloem parenchyma. Phloem parenchyma. Do you recall? Yeah, phloem parenchyma. Next, another thing I wanted to say. When we were talking about phloem at the time, when we come across fibers, observe, I said fibers are present in some kind of phloem, fibers are absent in another kind of phloem. Could you tell me which phloem fibers are absent, which phloem fibers are present? Exactly, where secondary phloem will have fibers. Okay, secondary phloem will have fibers, primary phloem do not have fibers. Observe the words like this. When you talk about phloem parenchyma, which is absent over here, in addition to that, there is one simple technical term. Observe the story. If you talk monocot stem, do they ever can have secondary phloem? Do they ever can have secondary phloem? Come on. No. Why? They can't perform secondary growth. That's why simply you can denote phloem fibers are also absent. Monocot fibers are absent. But when it comes to dicot, the phloem fiber word you have to clearly mention. Primary phloem, no phloem fibers. But when it comes to secondary phloem, phloem fibers can be noticeable. Okay? Next one. This is about vascular bundle and their scattered nature. This much information about monocotyledonous stem is more than sufficient. Then go to leaf. Observe, we have two varieties of leaves over here. One is dorsiventral leaf, another one is isobilateral leaf. First I will complete dorsiventral leaf, epidermis. Or if you go to comparatively dorsiventral leaf and isobilateral leaf comparatively, that would be easy to notice. Then let us take, this is dorsiventral leaf, this is isobilateral. Simple differences are there, but you have to remember those differences very clearly. See, when do you remember the differences? If you know about their particular location and particular function, you can remember them automatically. For that, you need to forcibly need not to remember them. That's why let us analyze. First one, dorsiventral leaf, isobilateral leaf, epidermis. This is adaxial upper epidermis and abaxial lower epidermis. Abax means Adax means these names were given based on the location of the plant stem. What I mean to say, you take one stem, this is the stem. This stem will have a flat body, the leaf. The leaf that facing towards the stem, this is called adaxial. And the leaf face away from the stem, that's called abaxial. Abaxial body is away from the stem, adaxial body towards the stem. Adax means attached. Here away from the stem. So abaxial surface is generally lower side. Adaxial surface generally considered as upper side. Apart from that, upper lower words may not be appropriate with respect to leaf, because leaf is a flat body. Sometimes some leaves will be flat, sometimes some leaves will be straight. For example, let us take a aloe vera leaf, well known thick leaf. Do you see aloe vera leaf flat like this? No, it will stay straight. The straight leaves, which are generally monocotyledonous character, that's why they are called as isobilateral leaf. The flat leaves, dicot character, they are called as dorsiventral leaf. So upper side adaxial surface, lower side abaxial surface. Then adaxial abaxial epidermis generally covered by cuticle. But one major difference is adaxial epidermis more number of stomata when compared to abaxial epidermis more number of stomata when compared to adaxial surface. Sometimes even adaxial surface, a stomata will be absent, but we cannot say all the time stomata absent in adaxial surface, but stomata is present, limited in number. Next one, go to isobilateral leaf. In isobilateral leaf, upper lower epidermis evenly stomata distributed evenly. You can see here there is a stomata, here also there is a stomata. That's why evenly stomata distributed. Since we are talking about epidermis, let us talk about the epidermal layer, especially in this area. In monocot epidermis I am talking, they have some special type of cells called bulliform cells. What cells? Bulliform cells. They have a special name called motor cells. Motor cells. What is the nature of them? They exactly present in adaxial epidermal surface of the monocot leaf, and they have one capacity, that is expansion relaxation. Expansion relaxation. The expansion of the cell, they known it as turgid. Turgid means expansion of cellular surface area. Turgid. When the cell become turgid, leaf open. When the cell become flaccid, leaf closes. What do you mean by leaf close, leaf open? Simple. Leaf, for example, in case of monocot, not all monocot this feature, only for some special grasses. Then they have, if it is a leaf, during night time leaf is going to curl inward. During daytime that leaf surface area opens. Curling in, opening up, that is due to presence of a special layer of epidermal cell all along the length of the vein. That special layer of epidermal cells are known as bulliform cells. If bulliform cells become turgid, the leaf surface area opens. If the bulliform cells become flaccid means loss of water, they are going to be close. I mean, leaf surface area is going to be close. One thing, bulliform cells characteristic feature to only monocotyledonous leaf. They could not be seen in case of dicot leaf. Next one, go to inside. The inner area of the leaf is known as mesophyll area. But when it comes to dicot, the mesophyll area divided into two parts. Towards the adaxial surface, cells are arranged like a palisade. Palisade means they are like a fencing, one nearby another they arrange. This is called palisade mesophyll or palisade parenchyma. Below the abaxial surface, the cells are arranged with large intercellular spaces. Such kind of thing is called spongy parenchyma. Presence of mesophyll cells in two forms, one is palisade, another one is spongy, is a characteristic feature of dicotyledonous leaf. But go to monocot, you can see in the monocot there is a existence of mesophyll area, but that mesophyll area is not distinguished into palisade, spongy. All over the area cells are compactly arranged, mostly with limited intercellular spaces. They will be arranged in the form of compact, but you don't find the distinction, which means you will find mesophyll cells both in dicot and monocot leaf. But in monocot leaf, these cells are compactly arranged without any intercellular spaces. But in case of dicot, they will be arranged in the form of large intercellular spaces containing areas. So this is the distinction in between the dicot and monocot leaf with respect to mesophyll area. Okay, then go to vascular bundles. Then first, If you are talking about vascular bundle, there is a difference in size of the vascular bundle in dicot and monocot leaf. Why? In a leaf, vascular bundles are found in veins. In a leaf, vascular bundles are found in veins. If you take a dicot leaf, veins reticulated or reticulate venation could be found in case of dicot. A dicot leaf with reticulate venation, different sized veins are found. As the size of the vein changes, the size of the vascular bundle will also going to change. Larger veins will have larger vascular bundle, smaller veins will have smaller vascular bundle. That's a characteristic feature. But observe in case of monocot. Monocots possess parallel venation. Because of the parallel venation, almost all veins are same in size. Same in size, almost all. I am not saying every vein will be equal to the size to another vein in case of the no. Almost all veins are similar in size. There may be a variations could be developed, but that variations are not that much significantly noticeable in monocot leaf when compared to dicot leaf. Then go to vascular bundle. Observe this. Vascular bundles will have bundle sheath cells. What is that, ma? Bundle sheath cells. Bundle sheath. Vascular bundles are having bundle sheath cells. What is this bundle sheath cell? As I said earlier, a covering will be present around the vascular bundle, that's called bundle sheath cells. The bundle sheath cells are parenchyma in the leaf. Parenchyma. Next one. You can see this bundle sheath is connected by some amount of parenchyma up and down. This is called bundle sheath extensions. Bundle sheath extension. Bundle sheath extensions. Okay. Above and below, there is a presence of bundle sheath. That bundle sheath extensions are also developed. Why bundle sheath extensions? These extensions keep the vascular bundle in the particular location. That means vascular bundle should be staying in the middle of the leaf. So there, the middle of the leaf vein should run. The vein will run middle of the leaf by having bundle sheath extension. Lower bundle sheath extension touch to lower epidermis, upper bundle sheath extensions touch to upper epidermis, so that the bundle sheaths will be keeping. Is it okay? Are you getting the idea? Bundle sheath cells are keeping these things to stay at a particular direction. Next one, go to monocot. Monocots do have bundle sheath extension. Monocots do have bundle sheath extensions, but the story is the monocots will have bundle sheath extensions because here we will find sclerenchymatous extensions. Sclerenchymatous extension. Is it okay? Now, this one has parenchymatous extensions. These are having sclerenchymatous extensions. Very important. That information not mentioned in the NCERT book, but you have to remember there are some areas where bundle sheath can elongate it in order to touch upper and lower epidermis. Next, another thing. Go to inside the vascular bundle. Anyhow, vascular bundle should be having xylem and phloem, but the location of xylem and phloem very peculiar in case of leaves. Remember, when we were talking about a stem, this is to be learned very, it should be learned very cleanly. Otherwise, you will get confused. Observe, this is a stem. This is the stem. Okay? In the stem, carefully watch what I am doing. In the center, I have xylem. That's common. Peripherally, I have phloem. Center xylem, peripheral phloem. Yes or no? When I am coming from the peripheral to inner, center you will have xylem. Center you will have xylem. Peripheral you will have phloem. I will take these bodies. Xylem I have kept red. Phloem I am keeping green. Could you see that? Now this green patch, and this is red patch. Now what I am doing, I will take a knife I will take, and I will exactly cut here. I have made a cut exactly in the center by taking a knife. Okay, now after taking that, I will bend it. This is the cut portion, middle transversely I am cutting, and I am bending it. If I bend this one, observe, if I bend like this, okay, this is the portion I have bent. After cutting, I have bent. Tell me, upper side which one will come, lower side which one will come? Do you understand what I am saying? I have made a cut in the center and I have bent it. Now, if I make exactly like this, this is a straight and I am bending like that. Okay. Now, upper portion what will come, lower side what will come? Come on, come on. Speak out to me. When I cut like that, okay, upper side what will come, lower side what will come? You have to answer. Say it wrong, say it right, doesn't matter, say something. Okay, upper side you will get xylem, lower side you will get phloem. Simple. This is lower side. I have made a cut like this, slowly bending. So this side is upper, this side is lower. Same thing will happen in case of leaf also. Leaf is a, do you remember? Leaf is a epidermal cell, apical meristematic cell modification. Apical meristematic cell only producing this leaf. Leaf is a cut portion of a stem, because stem only can produce leaves. Got it? If so, observe. Whenever we talk about leaves, carefully, whenever we talk about leaves, okay, upper surfaces, listen, whenever we take about leaf, I'm talking, this is a vascular bundle. Inside the vascular bundle, I'm talking. Okay? Always lower side, this is the place. You will have phloem. Lower side. Towards the abaxial surface, phloem. Towards the upper side, xylem. Xylem always towards the upper side, phloem always towards the lower side. This orientation never changes. My dears, remember that. Xylem always will be present towards the upper side, phloem present always towards the lower side. Why? I have explained there. Got it? Then come back to the story. Same thing arrangement they have clearly noticed in case of our monocot leaf. You see this. Phloem towards the lower side. Just they have mentioned a word. Xylem towards upper side. This is how xylem and phloem will be arranged. But when you talk about cavities present inside the xylem, maybe dicotyledonous leaf will have smaller cavities when compared to monocot. But the cavity, depending upon the size of the leaf, some plants produces large leaves, some plants produces smaller leaves. So the cavity may going to be varies depending upon the type of cell where we are depending upon the type of plant what we talking about. That's why cavities are not significant here. Understand? That's about dicot leaf and monocot leaf. If you have any doubts, you can post it there. I can clarify. That's about the 4 organs that what we have to learn with respect to dicot and monocot organs. Do you have any doubts? That's about today. Okay, next class we will start secondary growth. Next one. Secondary growth should be discussed. You may be getting thoughts that secondary growth has been deleted from the NCERT book, but here and there in an area called plant growth and development, they are talking about the terms like xylem differentiation, phloem differentiation, this phytohormone doing this differentiation work. Those words, if you want to understand, differentiation, de-differentiation, redifferentiation, etc., that you must be having some idea about secondary growth. That's why we will discuss in the next class secondary growth. Is it fine today? That's about today. Fine. Thank you. We'll meet in the next class.